Good day, ma'am. I am Danica Santos from the section of 338-1, and I'm here to present is all about the DNA analysis. So first is the lesson objective, which is to explain the importance of the DNA analysis and to enumerate the steps in the DNA analysis. So first is the DNA or the deoxyribonucleic acid, meaning ito yung mga self-replicating material which is present in all nearly living organisms such as tao or people, human, animals, and insect is that nagko-contain niya ng mga DNA. So, located ang DNA sa mga cell. And that cell contains the nucleus. So, that nucleus contains the chromosome which is identified for the DNA. So, ang nature and composition of DNA is that each nucleotide contains the phosphatate group such as the sugar group and the nitrogen group. So, meron ding 4 types ang nitrogen bases, which is the ATGC or the adenine, the thymine or the thymine, the guanine, and the cytosine. And that order of these bases is what determines the DNA instructor or the genetic code. So next is the forensic DNA analysis and how it is used to solve the crime. So meron dyan mga hair evidence, fingerprint, blood, saliva, urine, and semen, and that are all biological evidence that can undergo to the DNA at nagko-contain din yan ng mga cell. Lahat yan. So, unang-una is in cases there are suspect to be identified, a sample of that person's DNA can be compared to the evidence from the crime scene na nakolek. And also, the result of this comparison is may help to establish the suspects if it's committed to the crime. And also, in cases where a suspect has not yet been identified, the biological evidence from the crime scene can be analyzed and compared to the offender profiles in the DNA databases to help identify the perpetrator. And also, crime scene evidence can also be linked to other crime scene through the use of DNA database. So next is the steps in DNA analysis. So una-una is the extraction, meaning the extracting the DNA from the nucleus to release the content. And the quantitation is to determine how much cell or DNA was retrieved from the cell. And the amplification is to determination of the variation of the DNA test results or types from the specific region of the DNA. And lastly is the detection process, which is the DNA separated and passed through the laser. So next po natin is the types of DNA analysis. So una-una is the polymerase chain reaction. So etong method na to is widely used for the rapidly making millions of copies of specific DNA samples. So ina-allow din nila ang scientists to take a very small samples of the DNA. So ang example natin dito is ang swab test. So, nagko-conduct din yon ng maraming copies in a short period of time. So, mas easy siya to use. So, next is the short tandem repeats, meaning it is the microsatellite with the repeats 12 that are 2 to 7 bases pairs to the lengths with the human repeats, verifying among the individuals. And the short tandem repeats also is the most beneficial for the human identification. So, next po natin is the Y chromosomes. So, bigyan ko lang din po ng additional. So, kapag ka-female ka, nagko-contain ka ng XX chromosomes, and pagka-male ka naman is XY chromosomes. So, balik po tayo. Ang Y chromosomes market target, only male fraction of a biological sample. So, next po natin and last is the mitochondrial DNA. Okay, so ito po is the analyst allows the forensic laboratory to develop the DNA profile from the evidence that may be suitable for this analysis. Also, the empty DNA is that ina-analyze nila yung mga DNA in a different part of the cell. So, ano-ano ba yung mga part na yon na ina-undergo din nila for the examination. Also, it can include the hair shafts, bones, teeth, urine, and everything that is biological evidence that is found at the crime scene. So in that cell, okay, so mitochondria naman ang pinanggagalingan, unlike the DNA is that sa cell and the nucleus. So dito naman po is from the mitochondria DNA. So dun lamang po nagtatapos ang aking presentation. Thank you po.